Then one day I talked to Dr. Wim Ganti and I told, I don't want to do any of the conventional techniques because it is giving less conclusive results. And also it is really tough to track a mechanism if you use a conventional techniques. And also at the same time, all the other labs, they don't have those facilities. So luckily we have facilities to the animal facility. It's like a walkable distance. We can use whatever imaging. And also I want to share to the students, if anybody interested to screen any of your stuff, we are ready. We are happy to help you. Even our industries also, even Dr. Gulati also. If you are interested, we are always ready to screen any of the molecules.
Actually, I mean, one thing which I'll admire is a nice presentation, of course, and it's not easy to build that kind of infrastructure and the facilities which you have built in. So I commend you for that. Regarding comorbidities, hypertension, diabetes, and there are other situations. Out of these, particularly from India, which one you think will be the best target to handle it first? And the reason I'm asking is that there is a group of neurologists in around Hyderabad area. What they did was they selected few villages and just monitored blood pressure and controlled blood pressure, and the incidence of stroke has gone down in the last 3 years. They could monitor. So your thoughts on which one is the most important?
So in clinical setting, we have seen a lot of patients. They recover more if they are under comprehensive antihypertensive agents and anti-diabetic agents. Most surprising thing is, I don't know whether it is there is no mechanism, but like a post-stroke recovery is very good if somebody quits smoking. Nobody so far discussed about it or demonstrated, but if you see the clinical data, if you quit smoking, the chance of post-stroke survival after TPA is more than 50%. And it is only 20% with the hypertensive management and diabetic management. When it comes to India, there is a regional disease prevalence. So if you go to south, it is more like renal problems. And if you go to urban areas, more so recently I came across one article when I want to give a talk in India. I checked what are the stroke structure in India. So what I identified is all the urban people living in the urban have more strokes, and all the rural area people having more cardiovascular, like heart attacks and all those things. And also the problem is stroke you can easily prevent by changing lifestyle and everything. So I think in India, I think it's better to start, India needs a drug like yours because it is kind of more economical. Because if you suggest a thrombectomy, you need a lot of people in comprehensive unit. You don't get a lot of specialists here. You don't get a neurosurgeon, neurologist, and an interventional neurologist here. So I think we need a proper drug candidate. But targeting a drug candidate with diabetes is more better in India when compared to the hypertension. The reason I'm saying diabetes is, if the person is diabetic, they also have problem with the drug clearance because they end up with getting nephropathy, diabetic nephropathy. I think that is one thing. And another thing, there is a new class of drugs now prescribing SGLT2 inhibitors. They are well known to control the hypertension and diabetes. Yeah.
Okay, so I have a question for you and a comment what you asked, Dr. Klatia. So I think that diet-induced obesity is a very nice model to, because a lot of people, we have fat. When we become old, we like to eat, all junk food, obese conditions, you have increase in diabetes, all DBDB models, you are making knockout, but in human, we don't do any knockouts. So I think diet-induced obesity is a very nice model to understand the comorbid condition, and I'm sure his boss is working on that. So my question is, okay, you are working on TNSNC, so you show some of the mechanisms, very nice talk.
Thank you.
One of the mechanisms you show is TNSNC is promoting thrombosis.
Yes.
And that's my field, and you also show that this is also promoting inflammation.
Yes.
But TNSNC is an extracellular matrix protein, right? It interacts with so many targets.
Yes.
Not only TR4.
Yes.
So do you think that TR4 is the only target, or it also interacts with some integrins, which are present on the neutrophil, because you show some slides on neutrophils.
So, yeah.
And second question is, why you think targeting senescence is the best approach if it's going through TR4? Why TR4 antagonist has not been successful in clinical trials?
So, yeah, very good question. So, the first question, why tenascin C is having a role is tenascin C already is a marker for atherosclerosis in the humans. And it is also identified in the stroke patients, and it correlates with the TLR4 levels in the humans. So, there is a clinical evidence show that tenascin C have a role by interacting with TLR4 in atherosclerotic plaque and also in the large artery atherosclerotic strokes. And at the same time, it also have a role in hypertension because it induces cardiac fibrosis. Coming to the next question, tenascin C have several functional domains. We tested only TLR4 now. So whatever I'm talking about the last slide, which have independent of TLR4 activities, that might be through the other functional domain. So now we are checking the fibronectin, and EGFR is another functional domain, and also integrins. And also we are checking all the things, but we have to start somewhere. So we started here.
Thank you.
Yeah, thank you.
Thank you.
If you don't have
any question, just last question. Yeah, just please be brief actually, because we are running late.
Good morning, sir. Thank you for delivering nice talk. I am Ziaur Rahman from Naipur, Hyderabad. Sir, actually you showed one slide like you are also working on stroke-induced gut dysbiosis.
Yes.
So we know that stroke will cause gut dysbiosis. So how will target both together, gut TLR4 and brain TLR4?
So if you are working on stroke, you always try to protect the brain. So the first thing, I'll protect brain, then I'll come to the gut microbiome or gut bacteria. And also gut bacteria is very old now. We recently published a paper in the gut virome. So you have more virome in your body when compared to the bacteria. That tell the bacteria what to do. So we are manipulating the virome now. So gut bacteria is almost 2015 and 2020. So it's almost done. But the question for you is, we have a special prophages and all the probiotics with a very specific phages and very specific bacteria. If you maintain the balance between the bacteria, I think it helps a lot. So the probiotics is a key. But the problem is what probiotics if you take and how much quantity you take, there is no proper investigations. The problem with the stroke and the use of probiotics is that some of the bacteria can travel, or metabolites can travel to the brain. And we don't have that information how they are going to affect the brain. But overall, clinically, if you use probiotics, it stabilizes the gut.
Thank you.
Thank you.
Thank you, Dr. Bharat, for nice talk. Thank you very much. Can you, madam, introduce next?
Next speaker is Dr. Anil Kumar Chauhan. He is a professor and director of thrombosis and vascular biology in the Division of Hematology, Oncology in the Department of Internal Medicine, University of Iowa. Dr. Chauhan has a lot of honors and credit. He has been awarded many honors, he has 75 publications, and he has made numerous contributions onto how adenosine molecule contribute towards thromboinflammation. He has been awarded a lot of grants, which includes R35 Outstanding Investigator Award. He also has been awarded from NIH and prestigious Established Investigator Award from American Heart Association. So he has a lot of distinctions and honors to his credit. And I now invite Dr. Chauhan to give his presentation.
I can stay here, sir. So, thank you very much for the kind introduction, and I would like to thank the organizing committee and the chair and the co-chair, and especially all the students. So when we came at 9, we thought that it's only speakers. We have to give a talk to the speakers. Now we can see all young faces. So let's change the gear. Okay. So last 2 talks you heard about ischemic stroke. And 85% of the ischemic stroke are caused by thrombosis. So I am going to talk So I'm a basic scientist. So what I'm going to show is that I'm not going to change the world, by this thing. And the topic what I'm going to tell you is a nice story, which is by accident. It's not I discovered something. We were trying to do something and just it came out. So, and most of the discoveries are made by accident. So what you see here is this is the aerial view of the center of the University of Iowa. This is a building and my lab is somewhere here. So this is our University of Iowa campus. So in this slide, I would like to give an overview of the research program in my lab. So since you know that thromboinflammatory process contribute to coronary artery disease and stroke, so the major focus of my lab is to study the interface between thrombosis, inflammation, and the vascular biology in context of stroke and atherosclerosis. And a couple of years back, NIH started a SPAN network. It's new, it's only 3 years old. And the main aim of this network is to bring together basic and translation scientists to systematically address all the prior methodological limitations of cellular protections. You know that 95% of the neurological protections, I think there were 1,600 which were successful in the preclinical trials, but nothing, when they came to clinics, all failed. So they have developed this network, and this is the site. If you want to go into detail, you can see these things. And there are 6 centers which was funded by NIH for the first span, and one was Carver College of Medicine, where I'm the PI on that, with Andy Cleara, who is a contact PI, Yale University, Johns Hopkins, MGH, Medical College of Georgia, and UT Austin. So what we do is, suppose you have a drug, and if you want to go, if you can put your drug in this trial, and if your drug works in this thing, so these are 6 centers, and we test this drug in different models, comorbid models, and if the drug passes the efficacy, you can tell the NIH it can go for the clinical trials, fast phase. And if the drug does not pass, then we just cut it. So recently, like 3 years or 4 years back, so I tried to focus on a different pathway. And that pathway is how to understand the metabolic plasticity contributes to platelet function thrombosis. So what you see here is the platelet receptors, and the signaling pathways that contributes to platelet function thrombosis. And all the green what you see are the current antiplatelet targets. And in the white are all the antiplatelet targets which are under clinical trials and could be potential targets. So for example, we have a abeximab which targets alpha-2b beta-3, one of the major receptors on the platelet, and then clopidogrel, Plavix, which targets the ATP. And then we use aspirin. People get stroke, then they are put on the aspirin, or combination of aspirin and clopidogrel so that you inhibit platelet function. And this aspirin inhibits COX. So when we have so many drugs, why we want to test more drugs? Why we want to do, either we want to do a basic science, or there's some limitation with every drug. So I think that, so these are some of the studies which you can go and look at the literature that this aspirin and P2Y inhibitors do not translate into clinical efficacy in 1/3 of the patient at risk for coronary syndromes and ischemic stroke. And they are like more potent antiplatelet agents, which includes like abeximab. Yeah, I cannot see. Anyway, so are associated with bleeding complications. So one of the things is that if you have a very strong drug, which is antiplatelet, then it leads to a bleeding complication, and you don't want that. So can you fine-tune between thrombosis and hemostasis? So now I come to topic of pyruvate kinase. It is heavily studied for last several years, and it just came by accident. I started going into this field. So this is a basic biochemistry. You know that there's a glycolytic pathway, and the glucose is converted to phosphoenol pyruvate to pyruvate and ATP. This is the last step of glycolysis, and this is controlled by pyruvate kinase. And then pyruvate then goes to the oxygenate phosphate cycle. It's a TCA cycle, and by LDH is converted to lactate, lactate dehydrogenase, under anaerobic condition. So this PK pyruvate kinase exists in 4 different isoforms. PKR, which is present on the erythrocytes, which is RBCs. PKL, which is in liver and kidney. PKM1 is present in the heart, brain, and muscle, and the PKM2, early fetal tissues and most early tissues. And this PKR and PKL are encoded by PKLR gene, whereas PKM and PKM2 are encoded by PKM gene. So it's very important to note that unlike other PK that exists as tetramers, PKM2 exists as tetramer and dimer. So these are made of similar monomers, but the dimer has different activity and the tetramers have different activity. So why PKM2 and not other PK isoforms? So as you know that most of the processes associated with platelet activation response to injury, including whether it's a platelet aggregation, alpha-2 beta-3 activation, granule secretion, spreading of clot detection, you need energy. So like normal cells in the resting platelets, the platelets depend on both the oxyphos and the aerobic glycolysis for the ATP generation. And this is happening in presence of oxygen. Whereas in the activated platelets, they exhibit a high level of aerobic glycolysis, which is shown in here. So, and dimeric PKM2, as you can see, is a tetrameric, and this dimeric PKM2. This tetrameric PKM2 and the PKM1 drives the oxyphos, whereas dimeric PKM2 drives the aerobic glycolysis. And why is it known as aerobic? Because this is happening in presence of oxygen, and this was first observed by scientist Warburg. It's also known as Warburg effect in tumor cells. And this molecule is very interesting because in addition to having its glycolytic activity, it also possesses protein kinase activity that might regulate platelet signaling and pathways. And this dimeric PKM2 also interacts with transcription factor STAT3 and promotes inflammation by regulating IL-1 beta and IL-6. So as I told in the first slide that most of the processes involves thrombosis and inflammation, including stroke, heart attack. So I started thinking, so I was in a seminar, I started thinking, oh, this molecule is regulating inflammation. And if it regulates thrombosis, then it could be a very good target because you have to use one drug to inhibit both things, which happens after reperfusion when you give a TPA or a mechanical thrombectomy. So if there are a lot of biochemists here, so people will ask, okay, this OXPHOS pathway, it generates 32 ATP or molecule of glucose, whereas this lactate pathway generates only 2 ATP molecules of glucose. So why platelets prefer activated this aerobic glycolysis for ATP generation? So I looked at the literature and I found that despite low efficiency, the rate of ATP generation is 100 times faster in the aerobic glycolysis compared to the OXPHOS pathway. So this could be one of the reasons because when platelets get activated, it's a very fast process. You get an injury, you start having clots. So it's a very fast process. You require instant source of energy. Maybe that's a platelets are activating that. So as we know that because glucose is sufficient and if inefficient but accelerated rate of ATP generation might be well suited to fulfill the short-term and high energy demand of the activated platelet in order to form blood clot in a shortest possible time. And another reason could be that aerobic glycolysis regenerates NAD plus a cofactor which is necessary to maintain high glycolytic flux from top to bottom, which may facilitate faster glucose metabolism to meet the short but high energy demands of the activated platelets. So based on this rationale, we build up a hypothesis that limiting PKM2 dimerization or deletion of PKM2 will inhibit aerobic glycolysis and thereby platelet function thrombosis. So approach was first to use a inhibitor, which is a small molecule amylase 5. It's available from the companies make it. So we wanted to just limit the PKM2 dimerization, and this molecule binds between the dimers and stabilizes the tetramers. So now the PKM, when you give this molecule, very specific for the stabilization of parameter. It does not interact with other molecules. This has been already shown. My lab was not the first to show that. And then we used the platelets, the genetic model where we just delete the PKM2 from the platelets to show that this is indeed a phenotype of the platelets and it's not an off-target effect of a drug. So first important thing is that what is the expression of PKM2? So we take the human platelets. So these are Western blots. 1, 2, 3 are three different individuals of 3 different mouse. And you can see a nice PKM2 expression both in the human and mouse platelets. And we use beta-18 as a control. So which question was, so this is all hypothesis. So we wanted to understand in platelets whether there is indeed there is an increase in the dimerization of PKM2 on activated platelets. So what we do is that these are resting platelets, and these are platelets which are treated with conluxin and thrombin. These are, I showed you, conluxin activates the GP6 pathway and thrombin the PAR receptors.
And these are different concentrations. As you can see here, between this resting and activate, you can see there's a nice increase in the dimer tetrameric ratio, which is quantified here, and each point is a single sample from a human sample. And these are all healthy donors. And if you treat ML2 you can see there's a decrease in the PKM to dimeric tetrameric ratio, as you can see here. So this shows that dimeric PKM2 is upregulated in activated platelets, and if you use ML2 which stabilizes tetramers by limiting PKM2 dimerations, you can limit PKM2 dimerization in the platelets. So this cartoons, so we shows that we want to test if you limit PKM2 dimerization, whether there is a decrease in the glucose uptake, and also there is a decrease in the lactate production. These are the results. As you can see here, this is a resting platelet, and this is the platelet-activated convolution thrombin. 2 different doses. Each point is a single human sample. As you can see that if you ML2 it emits glucose uptake in a dose-dependent fashion. Also, there is a decrease in the lactate production in the activated platelets when you activate with convolution of thrombin, suggesting that ML2 by limiting PKM2 dimerizations inhibits glucose uptake as well as there is a decrease in lactate production. So, obvious question is, if you stimulate platelets with agonist, and then you pre-treat platelets with this ML2, whether you see any inhibition. So what we do is you take you isolate platelets or platelet-rich plasma, pre-treat with the inhibitor at different doses, and do a platelet aggregations with a agonist, which is GPC agonist like convection collagen, and GPCR agonist, TRAP and ADP. On the top, it shows the aggregation profile. On the left-hand side is shown the aggregation. Each point is again from a single human donors. As you can see, that percent of platelet aggregation is inhibited with ML2 in a dose-dependent fashion, both with the GP agonist, convection collagen, and GPCR agonist, suggesting that limiting PKM2 dimerizations inhibits integrin inside outside signaling. These are some of the markers on the platelets. So we want to look at whether inhibiting PKM2 dimerization also inhibits the P-selectin, which is alpha-2 P-tetra activation, or alpha granules or dense glucose secretion, which is P-selectin and alpha granules. As you can see here, that we use 2 different doses, and it's also dose-dependent fashion. If you inhibit, the human platelets which are treated with ML2 they have a less PEC binding, which is decrease in alpha-2 B3 activation. They also have less CD60P fluorescence, which is the P-selectin secretion as well as the ATP generation, suggesting that ML2 inhibits alpha-2 B3 activation, alpha and dense glucose secretion in human platelets. Next question is that whether the ML2 is also inhibiting the integrin outside in signaling. So we went for a clot retraction assay, and you can see this is a vehicle. This is ML treated with 50 micromolar, this is with 100 micromolar, and this is on the left-hand side from the quantification. This is with the vehicle, black is with the 50 micromolar, and this is with the 100 micromolar. As you can see here that the clot retraction was significantly inhibited in the human platelets which are treated with ML2 after thrombin stimulation, and this was dose dependent. So this is all good because we are using in vitro situation, in vitro because, and we were using platelets, purified platelets or PRP, which is platelet-rich plasma. But in real situation, you have whole blood and the other cell types. So we went for another assay, which is a microfluidic chamber assay, where you coat with collagen and then you pass the whole blood. And the beauty of this technique is that you have parallel channels, and one channel you can put a vehicle-controlled pre-treated whole blood, and this is ML2 treated whole blood. And then you can quantify over time. So as you say, this is you can quantify the thrombus growth, and this is a percent of area covered by the platelets. As you can see here, again that ML2 inhibited percent of area covered by the activated platelets, and also there was decrease in thrombus growth size, suggesting that by limiting PKM2 dimerization, you can inhibit platelet aggregation in vitro in whole blood. Then we went to look at the mechanism, which is because I told you that PKM2 also has a protein kinase activity. So we look at the mechanism. So here you show this is the resting platelets, and these are the platelets treated with convulsion and thrombin. As you can see here, the amyloid 5 inhibits phospho-AKT as phospho-GSK3 beta. And here is a quantification. You can see that at 2 different concentrations, amyloid 5 inhibits both the phospho-AKT and the phospho-GSK. And we use total AKT and total GSK3 beta as the loading control. This is very new, it's not published. So we went and looked at the metabolomics on the global snapshot. And we just used 3 samples because it's run on triplicates and 1 sample costs $500. So we look at, so we don't have so much fun, so I just wanted to start. Now I got some money, so we are going to continue this study. So this is a global snapshot of metabolites. This is with the resting platelets, this is with convulsin, which is agonist for collagen, and this is inhibitor. You can see all these things. But then you have to use a tool like gene analysis, where we found that inhibiting PKM2 dimerization with amyloid 5 inhibits glycolysis, glycerol phosphorylate metabolism, as well as arachidonic acid metabolism, which is the formation of thromboxane. That's one of the mechanisms how platelets get activated. And this is the quantification from 3 different individuals. Of course, this is all preliminary data because this is just 3 individuals, though it's significant, but we are going to repeat this thing with more than 20 samples, and we are in the process of working with the core facility to generate more data. So until now, what I have shown is that if you limit PKM2 dimerization by amyloid 5, it reduces glucose uptake, it results in reduced lactate formation, somehow indirectly or directly, we don't know the mechanism. It inhibits the phosphorylation of PI3 kinase. It also inhibits the phosphorylation of AKT, and upregulates the GSK3 beta, leading to reduced platelet activation, aggregation, and thrombosis in human platelets. So thrombosis is not a correct word to use here because what we are doing is all in vitro, because it's not possible to do in vivo. So once these studies were confirmed with the human platelets, then we did same study with the mouse platelets, and we found the similar results with the mouse platelets. So because of less time, I'm not going to show the data with the paper, but it's all published, so you can go and see the papers. So obvious question is when you are using inhibitors, the first question people ask whether it has an off-target effect. So we went and made we crossed the PKM2 flox mice with a PF crow mice, and this is the PKM2 deficient mouse, which is a platelet-specific PKM2 deficient mouse, and this is the wild-type controls. It's important to know that all the studies in my lab, we use literate controls. So this is a Western blot to confirm that there is no PKM2 expressed in the platelets of a PKM2 deficient mice. And this is an additional control to show that when you delete PKM2 only in the platelets, there is no deletion in the macrophages. So this is a cartoon showing that here now, because it's not possible because PKM2 had 2 different isoforms, and at this point, I did not have the tools to just remove the dimeric PKM2, so we have to just remove both the isoforms of PKM2, which is dimeric and tetrameric. So whole idea was that if we are deleting both these things, whether we can see a decrease in the glucose or lactate production or not. If not, then this is not a nice model. So we went and looked at the lactate production, and also glucose is already published. I just put this thing. So this is the resting platelets. You can see there's no difference. But if you delete PKM2, there's a reduction in the lactate concentration. Though that is significant, but it was not so strong effect with what we see with the human platelet. However, this suggests that this mouse model can be used to test the effect of PKM2 on platelet function thrombosis. So then we did the platelet aggregation studies. And I would like you, so on the top are aggregation profile curves from the wild type and platelet-specific PKM2 deficient mouse. And I would like you to concentrate on this red things. So when you use a higher concentration of convulsin, we did not see any difference. So this was surprising. But when we use suboptimal concentration of agonist, there was a very strong effect, suggesting that if you inhibit PKM2 dimerization or PKM2 with a suboptimal concentration, so if you use a suboptimal concentration of agonist, then you can see a defect in platelet aggregation in the PKM2 deficient mouse. Again, this is the same assay where we look at the